morning. Thank you everyone for being here. A big pleasure. Thank you, the organizing committee, for this amazing scientific program. Every year, I think that is with the projection, or after a few years, it's getting a very high scientific quality. So I feel very honored to be here and to make you a summary of our results. When the scientific committee contacted us for giving a tentative title, I was thinking about, okay, let's try to talk about just focusing on new therapeutic perspectives. But as you will see along the presentation, during these last months, we have been also working on different fields, on precision medicine, on functional annotation, improving or adding some types of GRIN variants annotations, and also developing, and Mireia has been doing, and her team, a great effort to increase the GRIN database variants. So I will try to compile all this data in less than half an hour, and to do it in a lay manner. So I'd like to start by giving this overview of what is the brain, or what is the physiology of the nervous system. We are focusing on GRIN-related disorders, okay, and the NMDA receptor. So I will stop because I'm shaking a little bit. But I would like to put this in a context of physiology. As you know, we are part of our genes are encoding for the receptors. Then we have been hearing these amazing presentations about how these pennies, how these different scents are making a joint effect. They are creating neuronal networks and brain circuits. So we have more this, at the end, this holistic view of how is a human being, a GRIN individual, or a GRIN children. And we really have to keep in mind all this complexity. So when we talk about GRIN-related disorders, GRINopathies, this is a new slide that I would like to illustrate, or to make some image about how we see GRIN, or GRIN children. They are just stars, all different, all brighting, and they are in this universe. So that's beautiful, and that's life, but that makes things very complicated for researchers, yeah? Because we need to understand how the different stars are working, behaving, and also to try to find cures for all of them, not just the more brighting or the more prevalent, okay? So I think that this is what we have been doing in the last year, trying to take and considering all these orphan variants, and not only the most prevalent. And that is related to getting a bigger repertoire, so characterizing all the different mutations, and we and many other groups are doing this, but also to find treatments for all of them. This is what we call the precision medicine. So this is, well, we have been hearing Andrew's presentation about, and very clear and very didactic one, about how GRIN variants are affecting NMDA receptor activity. In this video, you can see, and this is a 3D model of the NMDA receptor structure that has been developed by Mireia's team. And this is the first version of all the variants that are present here. A variant is a change on the nucleotide that sometimes it can have pathogenicity that is associated, but sometimes it's just neutral. It's just like normal variation, okay, in neurotypical individuals. So as you can see in the video, there are some spots that are in red. That means that these positions are very sensitive, and individuals that are harboring variants in these positions, they have clinical manifestations, and they are GRIN children. So roughly, we have been classifying these changes on the physiology of the NMDA receptor as loss or gain of function. But in between, and yesterday in the roundtable we were talking about this gray zone, what is in between, no? And this is an important issue, especially when we talk about finding treatments. This is somehow the outline of my presentation. I'm going to show you some advances on the functional studies and how we can identify the pathogenicity and to stratify, to put a tag, like this is a gain of function or loss of function, a complex one. What about this complex one, okay? I will also, of course, these functional studies, if we want to find treatments, we need preclinical models. So models that allow to understand the pathology, but also to test specific compounds. And I will show you in the last slides the clinical studies and the drug discovery that we are starting. So a few years ago, in 2021, we published the GRIN variants database that was compiling the data from different groups, all the reported variants that were published or that were in different databases, the one from Leipzig, the one from Emory, those from other research groups, also our data. So we were compiling this GRIN database server in which people was visiting. So currently this is an average of the visits that we are getting in this database that was built by Mireia Oliveira and Adrian Garcia. And at that time, there were about more than 4,000 variants. Not all of them were pathogenic, of course. And among these, only 444 were associated with the disease. So with the recently, there has been released the genome and genome analysis has increased a lot. So there are about 800,000 health individuals, okay, that have this exome that has been sequenced. And this information about, let's say, neurotypical or healthy population is also informative about how a change on a green nucleotide can be without an effect. So we have been integrating not only these genomat or neutral variants, but also the pathogenic that have been released. And we recently have released the version 2 of the GRIN database. So you can see additional details, but in brief, what you can see is that we mostly with this update of the database, there are we doubled the number of GRIN-associated variants that are pathogenic. So the GRIN variants, they have there are lots of different GRIN variants. The first question, of course, when families they get a GRIN diagnosis, genetic diagnosis, is to know whether this variant is the cause of the clinical manifestation or not. And second question is to know if this is a loss of function, or some families they say LOF or GOF, no? A loss of function or gain of function. But with the discussion with the members of the association, and especially with Amela, she was very challenging and she was very inspiring. She was also, I think that this is a point that is important when we come to this type of meetings, that we have the feedback from the families. And we open our eyes and we understand how important it is to stratify these complex variants, those that are just orphan variants in an orphan disease. It's really dramatic. So we have been doing an effort to try to classify these orphan GRIN variants that are including these variants that are the so-called complex variants, that they have some features of a gain of function, but some features of a loss of function, but also to identify which are the C-terminal domain variants that are also very intriguing, because for using Xenopus oocytes or even in HEK cells, the currents are electrophysiological properties are not that affected. because of the use of heterologous expression systems. And we have been also studying new variants. So this is how we proceed when we annotate the GRIN variants. This is well, some families have been visiting the lab. So basically what we want to measure is the communication between cells, how these NMDA receptors are being activated, and how they allow, or how the flux of the influx of ions is affected. So we are using this kind of electrophysiological techniques. And of course, we need lots of students and time. So this is just an analog picture to the students that have been participating. So traditionally, we have been characterizing the mutations in the lab using hex cells that are not neuronal, okay, and that they are very useful for primary screening of the mutations effects. But for the complex mutations or the complex variants, they are not responding completely to what is happening with these mutations. So to address this point and to annotate the orphan GRIN variants, we have started with the support of GRIN Patias, the Spanish Association, and the Italian Association GRI, to develop a new cell-based assay that is consisting on establishing a primary neuronal, well, a new in our lab, okay, establishing primary neuronal cultures and expressing these GRIN complex variants or orphan variants, and to characterize what is the global effect, the overall effect in terms of NMDA receptor-mediated currents. So using these different cell-based assays from the 367 requests, we have been annotated in terms of pathogenicity 353, and the functional annotations using the GRIN database or collecting the data that have been reported by other groups, we have been able to respond to 292 families. Okay, second point is about we have this universe of the GRIN variants. Our first dream was to, or is, and common dream, is to annotate all of them and to have like different categories. But second one is to understand what is the effect of these mutations in animal models and to test different compounds. So to tackle this important goal, we have been developing different models. Some zebrafish models we have in the room, Silvia Lokovice. Silvia is there. She has been making an amazing job. I'm just putting an advertisement. She's looking for a postdoc, so she's a brilliant student, hard worker, amazing person. And she has been doing, creating a library of zebrafish models that have deletions of the different GRIN genes paralogs. We have been also doing experiments in GRIN2A, GRIN2B, loss of function, the heterozygous models, and in a GRIN2A gain of function model. And of course, you can see additional details on the posters. So we have, with these models, we have characterized the phenotypes. I will be happy to discuss also some differences or some commonalities with other rat models or some other knock-in mouse models. But at the end, they are just tools to check or to evaluate whether a new compound or any compound can be useful. And the principle, the strategy for treating GRIN-related disorders is, I think that in this image, you can see this gradient. So NMDA receptors activity needs to be very well tuned. We have to be in this central zone in which this is what we call homeostatic, okay? This is like in the range of normal neuronal function. But when we move to the left, to hypofunctionality, or to the right, too much activity, then there is a misfunction. So what we are trying to do, and all the people that is working with NMDA receptors and in terms of modulating the activity of these receptors, is to compensate, to restore a normal activity. So I'm going to show you in the next part of the presentation the results that we obtained with L-serine and with novel molecules, new molecules. The first thing is that we wanted to identify not only L-serine, but additional modulators. We talk about allosteric modulators. Allosteric modulators can be negative or positive. A negative is one that is decreasing the activity of the NMDA receptor, and the positive is the one that is boosting the activity, okay? And they are allosteric modulators because they are binding to different parts of the receptor, not in the agonist binding site. Okay, so how we proceed? That's a tricky question. So because we know the structure of the NMDA receptors, but there are thousands of different molecules that could be used. So this is where Mireia was using her model. So in her structural model, she knows perfectly all the shapes, all the edges of this receptor, and she was looking at potential interactors, molecules that could be binding to the receptors and changing the activity, yes? So I try to represent these different molecules with different shapes, different colors, and one of the important goals is to find these allosteric modulators that could be perhaps specific for some kind of receptors. You know that there are GRIN-2A, GRIN-2B. We have very good presentations on that sense. And we have this lock and door binding. So some molecules, ideally, we would like to have molecules that are specific for one type of receptors and not affecting the other ones, and vice versa. So this is what we are trying to do. The computational design, sorry, this is in Spanish. We evaluate these compounds. We have identified, or in silico identified, the candidate ones. We have 12 different candidates. We are evaluating some of them in vitro, some of them in vivo, and then of course the final goal is to go to the clinical translation. So I'm going to show you the results on the preclinical screening for different molecules. We have right now 4 molecules. One is mostly robustly and is L-serine. So this is how we dream to do with many other molecules. And I'm going to also show you some clinical studies with proof of concept studies in one particular molecule. So with about L-serine, and I'm short on time, so this is how we were thinking about using L-serine. If we have a receptor that is somehow not, so a loss of function GRIN variant will provoke a decrease of the activity of the NMDA receptor. So somehow the rationale is that we want to favor the activation of this receptor to recruit more receptors, or at least to enhance the activity. So with L-serine, our idea was to provide L-serine to the kids. This L-serine is metabolized and is converted into D-serine, and D-serine is a coagonist of the NMDA receptor. So potentially, D-serine could be increasing the activity. And this is what we observed. So I'm going to skip the fine details that was published in 2019, okay? But basically what we observed is that D-serine in cells was increasing the activity of a general population of NMDA receptors, not only the mutant ones, but also the wild-type ones. That was interesting. We observed in head cells, in primary neurons, and then that was the key point of the study, and that was performed by Ángel García Carzorla and Natalia Juliá, that they were brave enough to start a proof of concept study, a clinical study, in which they were administering L-serine that was demonstrated previously to be a safe treatment for children with metabolic disorders. Okay, so they used the same dose and made a first study with Paula. And that demonstrated, that was just the proof of principle, N of 1 study in which we observed that it was a benefit in terms of communication and motor skills. And from this study, we moved to a clinical trial, the first clinical trial for GRIN-related disorders that was reported, at least, in clinicaltrials.gov. And we published recently, the study was performed in 23 Spanish patients and that were treated for 12 months with Alcerin. This was a non-randomized study that confirmed the previous results that we obtained in the proof of concept study. And importantly, it was also somehow replicated or confirmed by additional studies from the group of Lemke or Van Karnenberg in the Netherlands. So overall, we are happy that other groups have been using also the treatment, and they are confirming this study, this pioneer study. And currently, I think that Alcerin is the commonly used treatment for GRIN loss of function individuals worldwide. However, Alcerin, as you can see in the previous graph, the changes are not that big or that huge as we would love. Okay, so we are looking for additional compounds that could be more beneficial for GRIN loss of function individuals. And of course, there are many other GRIN variants that still need personalized treatment. There is the Radiprodil trial, but of course, this needs lots of different therapeutic tools in the drug toolbox. So we have been working with the same hypothesis. From the 12 different allosteric modulators that were previously identified by Mireia, we have been conducting experiments with AM1. AM1 has been showing, we'll have the poster also for more details. AM1 is increasing the currents. You can see this is in the black trace is in the absence or only with the coagonist. And when we treat these cells with the coagonist plus in the presence of the allosteric modulator 1, they increase the current. So this will be potentiating, okay? This is in HEX cells. We have also observed in primary neurons that AM1 is increasing the number of synapses upon a synaptic plasticity paradigm. And we moved to the preclinical model. So we were using the GRIN2B mouse model. This is a test in which it's super dark. We do the experiments on a dark situation. Okay, mice are a night animal, so they move and they are less stressed under dim light conditions. And this is how we evaluate their learning and memory abilities. So basically, they are on this square. They have to explore. This is our specimen. Yeah, this is going to be okay. It needs to explore different objects, and we repeat the test 24 hours later, and we change one of the objects, and the mouse naturally wants to explore the novel object. Okay, so this is the novel object recognition. If the mouse remembers more. If it has a memory deficit, it will forget and will not identify the novelty of the object. So we have been conducting these experiments. These are a large number of wild-type mice or GRIN2B mice. There have been the experiments also, and I think this is an important point, to discriminate between males and females, yes. And sometimes we detected some differences, but in this test in particular, this was converging effect. So we observed the same phenotypes, and then we pooled together males and females. And first thing is that GRIN2B mice, they had a discrimination deficit, okay, this is without the treatment. But after a couple of months of treatment, these mice, they improved the recognition. So they were no differently behaving as control mice. In this graph, I don't know why there is a missing part of the graph here. Okay, so trust me, this is what was happening with chemical LTP in terms of the currents when we try to have an electrophysiological view of the activity of the CA3, CA1 circuit of the hippocampus. We observed that the GRIN2B mice, they were deficient, and with the treatment, the same mice that were treated here for the behavioral studies, we used postmortem to identify this effect, and we observed that the differences with respect to the wild-type mice were abolished by the treatment. So overall, with this data, we translated to the clinical practice together with Angels and Natalia. We performed a first proof of concept study with AM1 prime, okay, a derivative of this. And this is the final result that we obtained when applying an adaptive behavioral scale in the two cases. So these spider plots represent the different dimensions, communications, daily life skills, playing, or whatever. No? The different, all those that you can observe here, socialization, and so on. And you can observe that this is on black, this is before the treatment, and after 12 months of treatment, the area was increased, meaning that these dimensions were improved after the treatment with this AM1 prime compound. Okay, so to end up, just a few words and very excited about this. As a scientist, I was thinking that, okay, well, this is not that 100%. I have no graphs to show you that this is like, you know, that's it. But we are very, very excited about these findings. We have started to characterize the effects of two more candidates. In HEG cells, we have observed that AM2 is potentiating GLUN2A or GLUN2B containing receptors. And for AM3, this will be acting as a negative allosteric modulator. We have preliminary data in HEG cells, okay? And especially we have been using the same primary neuronal cultures to see if the overall output of this treatment, the overall output in terms of NMDA receptor excitatory postsynaptic currents were affected. And we were confirming that AM3 in neurons is decreasing the EPSCs. So these are very exciting news, and I would like to summarize with a couple of, well, many conclusions and some pictures. So I don't know, for those that are here, if you can profit and see these Catalan human towers that are starting with people that they come together, they push and they generate a strong core, okay? And from this, people is adding and growing up. So at the end, we have very young kids. They are like 4 or 5 years old. They go up to 20 meters. These human tower castles, they can reach 10 floors, okay? And this is the idea of what, when we come here, we do have when talking with families, clinicians, and researchers, that this is really what we need, especially when we work on rare diseases. So to summarize, GRIN variants are diverse. Keep in mind this universe of stars. Our kids are our stars, always. We have released an updated GRIN database. We are starting the annotation of these gray zone GRIN variants, and several preclinical models, cellular-based assays, and animal models, the zebrafish and mice, have good endophenotypes to evaluate the efficacy of the compounds. The Alcerin clinical trial has been completed, and we have promising data on AM1, AM2, and AM3. So last slide, I want to acknowledge you, scientific committee, for organizing this amazing meeting, all the families for participating and your daily dues. and all the people from the lab. So this is also we are not that big. This is the unit of physiology. Okay, we are just few, but very talented students. So now, currently, Alba Rejano and Laia Lopez will be keeping her activity in the lab. From the University of Vic, Mireia Oliveira is a big part of this journey. And Adrian and Gabriel and Sergi also are super hard workers, very smart and very kind. So also people from the Hospital Sant Joan de Déu, Angels and Natalia are doing an amazing work, not only locally, but I think that all the clinicians that are in the room are benefiting from their expertise and they're sharing protocols and everything. And of course, Silvia Lokovici, remember, she's very talented. So people seeking brilliant postdocs, you have Silvia there that is getting red. So thank you very much.